Welcome, AP Bio students, to your review of cellular biology. You have a test coming up here soon. I wanted to hit on some of the points that I know are going to be emphasized on the AP exam, and this will also be helpful for your test that's coming up in class here shortly. So follow along if you will, please. There's going to be 3 points here I want to make real quick that I know will be emphasized on the AP exam. First, you should know that all cells are membrane-enclosed bodies of cytoplasm. That means that all cells on Earth contain this stuff called cytoplasm, which is similar in chemistry to seawater. They're enclosed in a lipid bilayer membrane, and those things are universally true of all cells. We know that cells differ in many ways, and plant cells and animal cells and prokaryotic cells are different in many ways from one another, but this is universally true of all of them. It doesn't matter if I'm talking about a bacteria or a fungus, or an animal or a plant or a protist. They all can be said to have membrane-bound enclosed bodies of cytoplasm. That's what cells are. Cells are the basic unit of life. All kingdoms of life have cells that have at least that much in common. The other thing you need to know is that this cell membrane that encloses all cells on Earth, it's selectively permeable, meaning that some molecules can move through it easily and some molecules can't move through the cell membrane so easily. The way that molecules move, we've got diffusion, we have osmosis, and facilitated diffusion. Those 3 are called passive transport, meaning that the cell doesn't have to use any energy to make these 3 happen. Diffusion and osmosis, that's just movement of molecules from high concentration to low concentration. Facilitated diffusion involves having to open up a larger door in the cell membrane. The door is made of a protein. And then the molecules are still just allowed to move according to just the random motion of molecules due to their kinetic energy. The last type of movement through cell membrane is active transport, and in that case the cell would have to use ATP to move the molecules through the cell membrane. Third thing I need you to know about cells is that prokaryotes are different than eukaryotes. You should know how they differ. It was told to me by students that took this class in years past that is heavily emphasized on the AP exam, that the difference between prokaryotes and eukaryotes. I would definitely make sure I was able to compare and contrast a prokaryote to a eukaryote. Prokaryotes lack a nucleus and organelles. They do have ribosomes because they need to manufacture proteins. But besides that, prokaryotes are very simple. This picture right here I like because it's a nice, neat little picture of all the different types of movement of transportation across cell membranes. So here's our huge cell. We have diffusion going on. That's molecules moving from high concentration to low concentration, and they're just shooting right through the gaps here in our phospholipid membrane. Keep in mind the phospholipid membrane, the phosphorus regions are hydrophilic, meaning that they'll hang out near water, and these lipid tails are hydrophobic, so they all want to orient towards each other. That's why we get this self-organizing lipid bilayer membrane, is just because of how the chemistry works out with water. Facilitated diffusion, again, doesn't take any energy on the cell's part. Molecules still move from high concentration to low concentration, but now through this protein door. We could also have active transport going on. That's where molecules are moving from low concentration to high concentration, and the cell is going to need energy to do that. It'll need these transport proteins, and it'll have to feed these proteins ATP to make them pump molecules against the concentration gradient. We also talked a bit in class about endo and exocytosis. I would make sure I know what that is. I would know where these vesicles come from, how they form. I'd know a little bit about Golgi apparatus and the endoplasmic reticulum, and how that all plays together. But I like this picture. It's a nice, neat little way to review the major points of transport across the membrane. I would definitely know about a diagram like this, and I would know all the pieces, parts inside a cell. I would also know if I showed you a diagram like this, like say on a test or something, I would know how to be able to identify whether it was an animal or a plant cell, a prokaryotic cell or a eukaryotic cell. This one is a eukaryotic cell. I can tell because it's got membrane-bound structures in the cytoplasm, whereas a prokaryote would not have any of this. They don't even have a nucleus. I can also tell that it is an animal cell because a plant cell would, in addition to a cell membrane, have a cell wall around the outside. Like I already said, I'd be able to name each one of these structures in here and tell me a little bit about what each one of these structures do. I'm probably going to ask you several multiple choice questions about what is letter A pointing to and what does letter A do, and I would definitely know those. They're in your book. You need to know them. That's that. These graphs, I would know about these graphs. Exergonic reactions and endergonic reactions. Exergonic reactions would release energy to their surroundings. They would have a negative delta G. Um, an example of a reaction that could be represented with a graph like this would be like a campfire, okay? Or hydrolyzing ATP would give you an energy graph that looks like this, because we'd take ATP and we'd make it into ADP, and that would liberate energy to the surroundings for the cell to use. Okay, and then we could have positive delta G reactions where we take low energy products, we reorganize the bonds and end up with high, or we take the low energy reactants rather and make high energy products by reorganizing bonds. That requires the input of energy. It gives us a positive delta G. That's the one that sort of breaks the second law of thermodynamics, but not really, because in a closed system or in an open system you can do that. We get sunlight pouring in, which basically allows us to add energy to our system and make higher levels of organization from lower levels of organization stuff. I would read a little bit about that and make sure I understood exergonic reactions and endergonic reactions. Be familiar with that. You're going to see these graphs on a test. Finally, that's an anti-gravity cat. I just think this is funny, because if toast always lands butter side down and cats always land on their feet, if you strap a piece of buttered toast to a cat, you end up with an anti-gravity cat. That will not be on your exam. I just thought it was an entertaining picture. I did want to remind you that I'm always available to help. If you email, I will do my very best to get back to you. Come in, ask for help. If I'm not teaching, I am available here to help you out. The email is rissi@westernschools.org. Feel free to email anytime. Good luck studying tonight. Have a good night.